I, Avinash, on behalf of Bionivid Technologies Private Limited, would like to welcome all the researchers and scientists who have managed to take time out of their busy schedule and be a part of this webinar. Well, for years, the research community has relied on platforms that use reverse transcriptase to convert RNA to cDNA. However, this entire RNA to cDNA conversion and amplification process introduces variability into the data, right? So, NanoString Technologies has come up with revolutionary platform called nCounter, which offers direct RNA detection of for robust reproducible performance and unbiased quantitation. I request all the participants to keep the app on mute. Otherwise I'll have to mute all the.
Can you mute everyone
I'll
instead of requesting? I think you should put.
Yeah, I'll do that. Yeah. So, like as I was talking about the nCounter platform as such, so the nCounter platform offers direct RNA detection for robust reproducible performance and unbiased transcript quantitation. So, like to shed more light on the same, we have with us the team from NanoString Technologies, as you would have already guessed by now. Mr. J. Manikandan, Mr. Marshall, and Mr. Ashish from NanoString Technologies are here to take all your queries, answer them, and shed more light on how can this nCounter technology help you in your research. I would also like to request all the participants to give. We would appreciate rather if they provide a real-time about the feedback. If they provide the feedback about the real-time application of this technology in their ongoing genomics research. Also, on behalf of Bionivid Technologies, as I have introduced her before as well, we have with us Dr. Roli Budwar. She's our Chief Scientific Officer, and she'll be moderating this session. From our end, and all the queries will be presented by her. So, without wasting much time, I would like to hand over the session to Mr. J. Over to you, Mr. J.
I think I'm on mute. Yeah. Very good morning to everyone. Can you hear me? Am I audible? Can you hear me?
Yes, we can.
We can hear you.
Yeah. Perfect. Yeah. Thanks. Very good morning, everyone. Thanks, Bionivid, for the opportunity to present the NanoString technology. This is the first time we're working with Bionivid, so we thought of doing an introductory seminar, both on the nCounter side as well as the new technology that we launched, Genomics Digital Spatial Profiler. So I will be doing the nCounter side, and then followed by Marshall will be covering the Digital Spatial Profiler. My name, Jay Manikandan. I look after Greater Asia Pacific business for NanoString. Been with the company for 6 years now, and I used to, in between, work with single cell technologies as well. And then most of my life, I was a genomic scientist for the last 20 years or so. So what I'm going to do is introduce the technology, nCounter technology, and some of the real-world examples where the technology is being applied. And then I'll take some questions probably in the middle or at the end after the DSP session. Hope it works. Oh yeah, it does. So the current challenges with the oncology research, I think you can broadly apply this to other research as well. The heterogeneity of tissue, the more the heterogeneity of the disease leads to highly heterogeneous response. That makes highly challenges to capture the response with the current technologies, and also the complex interactions between the tumor and tumor microenvironment poses a lot of challenges in the translational research. And also many targets and interesting pathways being studied these days. And this leads to large amount of data being generated. That poses a lot of challenges how you want to measure. And you cannot work with single molecule or single biomarker to give you an answer. So if we're dealing with we're talking about set of pathways, several pathways and several genes within each pathways, and trying to understand immune response or immuno-oncology response. Gene expression is fantastic, powerful tool in translational biological complexity. You can measure biological phenotype, and DNA is fantastic. You can do epigenetics, and you can do copy numbers and mutation analysis. But then, in many situations, you cannot measure, for example, immune response with the DNA. So you need RNA. So RNA, it perfectly makes sense to measure pathways versus immune response, and also therapeutic targets and analyzing pathways analysis. However, the problem with the RNA using the current methodologies, we're not measuring using directly RNA. We're with all the technologies that I'm going to talk more after this, but all the technologies that we're using right now uses measurement using complementary DNA, cDNA. So what you're starting with, and then after converting into cDNA using reverse transcriptase, the starting abundance of your RNA is not the same once you convert into cDNA. And then once you amplify, it further deviates. You start with 3 molecules of RNA1, you end with 10, for example. And then you start with 3 molecules of RNA2, you probably end with 6, and the other one probably 8. So your starting and your end is not the same. It poses a lot of challenges with the current technologies. This is mainly because the protocols are very lengthy, and the count that you are not quite accurate to that original count. So what NanoString tried to do is that, how can we measure RNA without any amplification, without any RT conversion? But if you talk about all the current technologies, in a qPCR, you have to convert into RNA to cDNA, and then you have to amplify and quantify. There are several steps involved along the process. Same goes to microarrays, converting to cDNA, of course, the lengthy sample prep, and then you finally hybridize and capture the fluorescent intensity, not really the actual count of the molecule. Versus RNA-seq, of course, you prep the library and then sequence it, but the library prep involves cDNA synthesis. And there are several steps. Some protocol takes a couple of hours, and some takes a week-long process, depends upon which technology. But then the more expansion of the workflow leads to more errors in the data. I'm not saying that one technology is better than the other, and all the technology that uses amplification are bad. But this is what we have. qPCR has been around since 1983 until now, 27 years or so. It's the technology we have. If you want to do discovery biology, you have no choice. You got to do RNA-seq. That's the technology we have. But it gives you an advantage that things that you cannot do with other technology. But at the same time, it gives you errors. So when you want to take that signature into validation, and when you want to convert that a transcriptomic analysis into a signature to bedside, or to give you an answer for a specific biological question you're answering, at that point, you probably need a highly sensitive, accurate, easy to use platform, which avoids all the RT amplification and easy to analyze data. That's what NanoString is trying to address with nCounter technology. Ideally, the gold standard platform would have no RT and no amplification, and directly digitally counting nucleic acid, and the hands-on time that you take from sample to data, and then how few steps available, from sample to data. And then is there a way we can multiplex, several genes or multiple pathways in a single assay without needing to add more sample input? And finally, of course, can you handle complex sample, highly degraded sample, FFPE sample, and things like that? Finally, how simple the data is to analyze, without needing complex bioinformatics and our hardware software infrastructure. That's, if you put together all of this, NanoString thought about how can we come up with a tool that address all of this problem. And that's exactly what they tried to do when they launched in 2003. A company out of Seattle, out of Institute of Systems Biology in Seattle. But our first system launched in 2008. We called nCounter Analysis System. And then a few years later, we also introduced a Sprint instrument, 2015. And more recently, in 2019, we launched our digital spatial profiler, which Marshall is going to talk a lot about that. To date, we have close to 1,000 installations, nearly 20 installations in India, over 3,200 publications. This is a little bit old slide, but I think should be more by now. Several, tons and tons of Cell Science, Nature, PNAS, high-end publications. I'll show you some of those case studies. People use the technology, but before that, the platform, as I mentioned, really designed for translational research. The only technology until today does not use enzymes. No enzymology, no amplification, directly digitally quantifying nucleic acid. This is really, really important if you're working with bad quality sample or FFP sample. The reason is when you have a bad quality sample and mix with RT enzyme, you're going to make, the enzyme is going to make complex. And then once it makes complex, then you cannot efficiently RT. And if you cannot efficiently RT, you cannot efficiently amplify. If you cannot amplify, you're not going to quantify. And that's the biggest problem with RT enzyme when you use with impurities or highly degraded sample. And because we don't use any enzymology, because our probes are directly complementing to the target of interest or your genes of interest, all we do is let our probes marry to your genes of interest and directly digitally count each and every single nucleic acid in your sample. So it's basically a counting technology using optical barcode. And then the count that you're getting is highly reproducible. We're talking about million counts per gene, anywhere between 10 counts to million counts. So the dynamic range is really, really long, very high, anywhere between 1 to 6 logs dynamic range. This is really, really important when you do in many technologies. For example, in microarray, after a certain point, the fluorescent intensity, if you sort by descending, you will see all the counts are same because they're all saturated for several genes. When you do a multiplexing assay, some of your genes can be highly expressed, some of your genes can be very low expressed. But the problem is in a single assay, if you put too much RNA, you're going to saturate some of the high expressed gene. If you put too less RNA, you're going to capture the highly expressed gene, but not the low expressed gene. But if you have a high dynamic range in a single input, you should be able to identify and capture all the high expressed gene at the same time, all the low expressed gene. And that's what Nanostring trying to do. Not many company talks about dynamic range because they saturate in those technologies. And then, more importantly, the ability to multiplex several genes in a single assay. Whether you want to screen one gene or maximum 800 genes using our technology, your input is always 50 nanogram. That doesn't change. The number of pipetting steps for one gene versus 800 genes, always 3 pipetting steps. Mixing our 2 probes with your total RNA, and that's it. Let them marry together and count next day. And that's why the workflow is super, super simple. Basically, we send 2 tubes of capture probe and reporter probe separately, pre-multiplexed. Let's say if you want to screen 50 genes, 50 capture probes in one tube, 50 reporter probes in one tube. We send you 2 tubes, and then once you have your total RNA, mix our 2 tubes with your total RNA, hybridize overnight, and load it into the system next morning. The system gives you data automatically after 200 hours. So everything is automated. And all you need from your lab to do an Nanostring experiment, of course, you need an instrument somewhere, but you need a pipette, 2 gloves, your total RNA. That's all you need. Nothing else required from your side. Like I mentioned, if you qPCR, I love the technology. It's great technology. If you're looking at 1 or 2 genes, I think you're better off using qPCR. You don't need Nanostring. You don't need microarray or NGS. But if you want to do discovery biology, obviously you need to do NGS and find all the novel genes that you're looking at, transcripts. But if you know set of genes through your microarrays and NGS or from publication or from qPCR data, and you wanted to validate directly digitally quantify those nucleic acids in a highly precise, quicker, cheaper, faster way, precise way, that's where Nanostring plays a role, anywhere between 10 to 800 genes in a single assay. So the way the chemistry works is a 2-probe chemistry, a 50-mer capture probe attached to a biotin molecule. If you see a biotin, you know what it marries with streptavidin, which is on our cartridge gel coated with streptavidin. So this biotin will bind to the streptavidin for purification purpose. The other half is another 50 mer. There is no gap here. Continuously, we take 100 mer complementary region from your gene of interest, and we split into two sites. First 50 goes into capture probe, and the next 50 goes into reporter probe. And then we add a long synthetic oligo and label with optical barcode. 4 color, 6 positions. It's a 6.6 kilobase long. Each one is 1.1 kilobase. So really, really long and can be seen under microscope. Basically, we take images and then look for combination of barcode and count how many times each combination of same barcode available on the cartridge. So if you see blue, yellow, red, blue, green, blue, this could be gene A. Basically, the system looks for how many times gene A is present or the same combination of barcode is present, and then puts them in a table. Assuming this is your mRNA or gene of interest, this is the complementary capture probe, complementary reporter probe, and the optical barcode. And then we just basically count and put them in a table. This is for the RNA and DNA. For the protein, we take the antibodies of interest and attach with a synthetic oligonucleotide using a photocleavable linker molecule. This is really important. I'm not going to talk a lot about protein, but when Marshall going to continue this session, he will talk a lot about this. Basically, when you shine a light, this photocleavable linker molecule cleaves the synthetic oligo, and you can collect them. We know the sequence of this synthetic oligo, so we can basically use the capture probe and reporter probe to count the synthetic oligo, which is indirectly counting the protein. And this is the same technology which is used in our digital spatial profiler, which we can also use with nCounter technology for detecting proteins as well as simultaneously detecting DNA, RNA, protein, all three biology at the same time. So on day one, if you have sample, which is basically total RNA, all you're going to do is 4-5 step, mixing our two little probes from two tubes, capture probe and reporter probe, with your total RNA and let them hybridize overnight at 65 degrees Celsius using your PCR machine, and not for amplification, but for incubation. And the next morning, you load that sample into system, and system automatically washes and then fixes, immobilize onto the cartridge. And then continuously the system will scan and gives you data straight away. But for a scientist, all it takes is only 5 minutes hands-on. That's all to detect 800 genes in 12 sample, in each sample 800 genes. So when you get the data the next morning, basically it will be each samples on columns, each genes on rows. It depends on which panel you're using or how many plex that you're doing, you'll get that many rows. Basically, it gives you absolute counting of nucleic acid from your sample. So you may have a normal healthy versus diseased, healthy versus cancer, infectious versus non-infectious sample. Basically, you're comparing two sets of data or multiple parameters and trying to find ratio, fold change, p-value, FDR, and so on and so forth. Of course, it comes with a free software called nSolver. It does all of these plots, clusterings, heat maps, even pathway analysis, advanced analysis you can do with the free tool. Of course, single platform. As long as you can barcode, you can count anything, no matter whether it's RNA or DNA, microRNA or protein. You can do all of these simultaneously, all of this together or separately, as long as you can barcode this. So we have slightly different chemistry for microRNA. And then, of course, you saw how the protein chemistry works, linking synthetic oligo to the antibody and then cleaving them and counting them. DNA is a little bit slightly different. Basically, DNA is super long and double-stranded. We break them into pieces by restriction enzyme and then denature at 95 degrees Celsius. Then the DNA becomes single-stranded and treated like RNA. The capture probe, reporter probe, and count the copy number variations. I'll skip the digital spatial profiler, leave it for Marshall. There are a number of panels available, off-the-shelf panels, if you go to our website, mainly addressing cancer biology, looking at pan-cancer pathway panel, immune profiling panel, and progression panel. There's also 360-degree panel looking at pan-cancer, immuno-oncology, and breast cancer, which comes with a data analysis report for these two panels. There are 15 different signatures built inside this panel, including IO signatures. And there's also lung and leukemia panels, myeloid, innate immunity panel, neuropathology, and autoimmune profiling panels. That's on the gene expression, but you also got human, mouse, rat, microRNA, copy number panels, several protein panels, and single nucleotide variants for solid tumor and heme. That's not all, but you can also customize any genes of your interest for any species. RNA is RNA. As long as any genome is sequenced by you or publicly available, you can share the sequence and we can design probes. We've done for hundreds of thousands of species, ranging from animals to plants, fungi, bacteria, virus, you name it, we've done it. Lots and lots of microbial species, plants, tons of plant examples. I'll show you a couple of them today, but of course, there are a lot more. I just want to switch gear and showcase quickly where the technology is being used, and then happy to take some questions now or at the end of the session. When the technology was new, a lot of people were skeptical and trying to benchmark against other existing technology. This is one example where Mayo Clinic in the US trying to compare nanostring data against Illumina sequencing data. Illumina was new at that point, launching RNA-seq. Same time nanostring launched just before that nCounter. And then they were trying to compare. And what they found is the correlation between nanostring data and Illumina RNA-seq data was fantastic, 0.97 correlation. And at that point, they bought our first system. And then today, Mayo has 6 nanostring instrument. All of their discovery biology come from Illumina RNA-seq, and then they make a panel, validate, and take it into clinic through nCounter. And in Broad Institute, they did slightly different. They compared RNA-seq, that blue line that you see, and nCounter green, and microarray. For several genes, they were trying to validate 3 different technology for 295 genes. What they found is in every single gene, RNA-seq and nanostring is precisely same trend versus microarray was completely different and very low signal for all of these genes. And just because the digital versus digital counting, both RNA-seq and nanostring. This is my most favorite publication among the 3,200 publication. In year 2012, Balco, he was a postdoc in the US, trying to use several years old FFPE sample, highly degraded, stored at room temperature on the desk. But he wanted to query 355 genes from each of these FFPE blocks. But the problem is, all he has is only 25 nanogram total RNA from FFPE sample. And then if you look at the electropherogram for all of these samples, there are no 2 peaks, which means the RNA is completely degraded, highly fragmented. I've never seen a Nature Medicine paper showing several electropherograms. Usually, high-end papers don't show all these electropherograms, whether there is 2 peaks or not. But in this paper, intentionally, they showcased that he specifically took those FFP blocks and trying to use 25 nanogram to query 355 genes. But what he did is, after run the nanostring assay, what he found was the correlation between one sample versus other sample for 355 genes. The lowest was 0.99 correlation, highest is 1.0. And this went into a Nature Medicine paper, basically claiming if you're working with clinical sample, FFPE sample, highly degraded sample, and do not, basically purposing, do not use any enzymology-based application like qPCR, microarray, and NGS, because you're not going to get data. If you were to use if you start with 25 nanogram, you can do 5 genes in a qPCR. You're not going to do 355 genes. So that's the power of the technology, and it's probably why the paper went into Nature Medicine. And after this, the scientific community realized that this technology is great for degraded sample, clinical sample, and FFPE sample, and we started seeing more and more FFPE users using the technology and surge in publications. Another great paper using FFPE sample, highly degraded sample, This is from Toronto, Sick Kids Hospital in Toronto, Canada. What they were trying to do is subtyping for brain tumor, medulloblastoma. There are 4 different subtypes. This is very important. Then you need to identify the subtype so that you can treat the kids with brain tumor appropriately. The treatment modality is completely different for each subtype. The original signature came from Affymetrix, but the problem is you cannot use Affymetrix in clinic. The results are not reproducible when you use FFPE sample. The experiment takes week long. So there are a lot of challenges. It's amazing. They did the discovery biology with Affy, but they could not take it into clinic. And they came to us, we have 23 gene that can subtype into 4 different groups that guides the treatment, but can you design probes? We designed probes. They did the experiment in Toronto, Canada, got nice separation of 4 different subtypes, precisely same as Affy. And then they didn't believe us, and they went to Miami, USA from Canada, redid the experiment with a different set of nanostring reagents in a different instrument, different person running in a different country. Again, got the same result. They didn't believe us. They went to Geneva, Switzerland, different continent, and trying to reproduce same result, exactly got mirrored signature, and published this paper. Before publishing, they went on to patent the signature, but then the charity that funded this money sued the Toronto Sick Kids Hospital. This is my money, and you're not patenting. Make it into public. Make sure that every children in the world uses gets benefit out of the signature. And then they decided to withdraw the patent application, put it in public domain. And then I literally took this signature from US to Asia, to China, to Thailand, to India, Australia, Malaysia, and Singapore, and people purchased the instrument just to do medulloblastoma and trying to identify the subtype and treat the children's. This is one example here. We work with the Professor Chitra Sarkar, Dean of Research in AIMS, Delhi. So I went to her for the first time. I trying to introduce the technology, and she was saying that she does a lot of medulloblastoma in India for children. She's a neuropathologist. She gets like more than 100 cases per year. But the problem is she takes 1 year to subtype medulloblastoma using qPCR, because there are 23 genes. You need triplicate, and there are housekeeping genes, and all of them are FFPE sample, and there are challenges in getting the precise result. And then she has to use another technology to make sure, probably immunohistochemistry or other technology, to make sure that the results are correct, because she's always worried whether you're getting the right subtype or not. And then I challenged her, give me 48 hours. I will do 96 sample, 4 cartridge per day, 48 sample per day, 2 days, 96 assay. We will do it. I'll prove this to you, and you can benchmark against your immunohistochemistry and previously done qPCR. If the results are same, will you buy the technology? And she said yes. And then we did this in just 2 days, 96 sample, which she was usually doing in 1 year time. She liked the technology, and the results are 100% correct, matching our existing result. And we sold our instrument, and of course AIMS is buying 2 more right now. So that the story behind medulloblastoma. The good thing with specifically in this story, the kids living with brain tumor, if you know the subtype, if you treat them appropriately, they live forever. They live 100 years, no problem, unless they have other problems in life. Unlike other cancer, extending life for additional 6 months is a big thing. In general in cancer world, but in these kids lives with brain tumor, once you treat them appropriately, they're fine forever. It's so good to see that it's happening in India using nCounter technology. Lots of pharmas uses our technology, hundreds of pharmas. You'll see hundreds of publications on the website. This is just one example. AstraZeneca using a nanostring and using FFPE sample. So what they say is, we found practical aspect of using nCounter platform favorable over any other technique used. Usually when pharma chooses a technology, they don't just go and decide, okay, I'm going to do nanostring, or I'm going to do RNA-seq, or I'm going to do qPCR. In every single time, they use all the technology, and they do the platform evaluation and decide which technology to use for each project. And that's, you'll see so many pharma publications. People used all the available technology, and finally decided to work with NanoString. And this is one of those studies. The study demonstrates that NanoString offers several key advantages, including sensitivity, reproducibility, technical robustness, using utility for clinical applications. You will see over and over again so many pharmas making this statement. The next story is another phenomenal story. In breast cancer, year 2000, couple of years before that, AFI launched gene expression microarray. And in 2000, at that point, people were using protein-based markers for cancer. And Chuck Peru, for the first time, trying to use FFP sample and use AFI microarrays and find molecular-based subtyping of breast cancer. It was never done before using RNA. And he did that. But the problem is the signature is too big, 8,102 genes. These are FFP sample. They could not use Affymetrix in the clinical setting for subtyping. It doesn't work, not reproducible. So Nature paper, amazing, but they could not use their own signature for next 9 years. In 2009, when NanoString launched the platform, they came back to us, can we use your technology? We said yes, and then they selected 50 genes out of the 496 differentially expressed genes. And those 50 genes they called FAM50. And then in 2000, they validated and republished a paper. And we liked the signature from our own customer. And we licensed in the signature. And then we did the validation for next 3 years in 3,407 FFP samples in multi-country. And that signature got FDA approved in 2013, 3 years later, both for the assay as well as for the instrument. To use it, FDA 510K approval, you can use a decentralized lab, which means anywhere in the world you can put the instrument and run the assay, versus all the other prognostic breast cancer assay today available, or only one lab is allowed to use. You need to send your sample to that particular lab and run the assay, versus Prosigna, you can have the instrument anywhere in the world and run the assay. You can imagine this, we're talking about 2020. 20 years ago, someone published a Nature paper, couldn't use the signature for 9 years until NanoString launched the first instrument. And then from 2009, it took another 5 years to get the FDA approval. In the last 5 years, NanoString is selling. Right now, it's under Veracyte, selling Prosigna. You can imagine the time it takes. If you don't have the right platform, the amazing Nature paper going to be sitting in the PubMed somewhere, right? It's not so useful, but it's amazing paper unless you use the right tool, right validation strategy, and right commercialization strategy. This is one of those examples. And then probably my last story before a couple of more plant example and wind this up. So another nice story, in the US, the 10 different hospitals made a consortium called Leukemia, Lymphoma, Molecular Profiling Consortium. Basically, what they want to do is find out the lymphoma subtyping, similar to the other medulloblastoma breast cancer subtyping. But the problem is, the existing methods they use is using immunohistochemistry based, which gives a lot of false positives. The original signature, 23 gene signature, came from Affymetrix, what they call gold standard, years ago. Again, same problem. FFP sample, you cannot use Affy because the results are not reproducible. It's a week-long procedure. You cannot put Affy into clinic and run the gene expression assay. And then immunohistochemistry gives 17% of the time false positive, which means there are two types of lymphoma. One is the aggressive form, which is activated B-cell lymphoma, which means patient's going to die faster, versus the other one is milder form, germinal B-cell lymphoma. If you're using immunohistochemistry, which is widely used currently, and now people are changing to nCounter-based Lymph2CX test. If someone is activated B-cell lymphoma, and if you're using immunohistochemistry, the IHC says, because it gives lots of false positive, it says it is germinal, then the doctor thinks, okay, he's fine, he's having milder form, sends the patient home. He goes home and dies because it's severe form. If someone is germinal B-cell lymphoma, and the IHC predicts as activated B-cell lymphoma, at this point, the clinician prescribed aggressive drug, which cost $150,000 per patient in the US, which is, the insurance company pays unnecessarily. That patient should not receive that aggressive drug So regardless, he's going to die because of side effects. It's going to die in 6 months. So 37, 34, if 100 patient goes to hospital, 17% of the time misclassification, 34 people going to die in 6 months by misclassification. So they came to us and they validated against nanostring. What they found is they used 50 cases. Only one case was different from AFI, gold standard. We don't know AFI was wrong or nanostring was wrong, but we took the blame that we misclassified one case and remaining 49 cases are same as the AFI gold standard and validated by other technology. And they published a one-page bullet paper. This was in 2014, January. And we liked the signature. In 2014 May, we licensed it from our own customer, which is from the consortium. Within 1 month, Celgene, biggest pharma in the US, came to us. I will pay you $45 million. Can I use the signature? And we said, fine. We took $45 million from Celgene because they have a magic blockbuster called Revlimid. If for Celgene to sell Revlimid, they need a precise test, companion diagnostic assay that correctly predicts which patient is activated B-cell lymphoma and which patient is germinal B-cell lymphoma. If someone is activated B-cell lymphoma, they will prescribe Revlimid. This is really important to sell more drugs because what Celgene does is put several instruments across US and globally and trying to qualify as many as possible activated B-cell lymphoma patients so that they can sell the drug. For clinicians, no problem. For patients, no problem because the insurance company pays $150,000 US per patient. Of course, you can also use the technology for detecting pathogens. In this case, PNAS paper identifying several bacteria, fungi, viruses, and parasites in a single sample infected with all these pathogens, and using nCounter probes, able to identify every single one of these pathogens. Of course, the technology is also used in plant world, agriculture world. This is one example that we used to work with the Malaysian government. You probably heard recently, India buys a lot of palm oil from Malaysia, the largest producer in the world. But the story began in Malaysia. The palm oil trees. You don't know whether the trees, the gender of the tree, whether it's male or female, until the tree grows, and then you see no seeds. Then you figure out, oh, this is a male plant. So what they wanted to do is, when the plant is young, they want to take the seed or root or leaf or stem and extract total RNA and run the Illumina RNA-seq, and found out that there are unique signature found only in the female plant. There are unique signature found only in the male plant or RNA transcript. And they came to NanoString. Can you validate the signature? And then they validated. They found they were able to validate the RNA signature data from Illumina-based RNA-seq to NanoString. And today what they can do is make a small panel. You don't need 48 RNA transcript to find it's a male or female. You don't need 48 from male. You can take a subset of this female-specific transcript and male-specific transcript, run a NanoString assay, and find out when a plant is young, whether this is going to be a male plant or female plant, and plant only those plants and throw away all the male plant. In this case, you don't have to wait 10 years and figure out, oh, this is a male, and you need to cut them up and and put a new plant there. So it's a very interesting study that we did. It's published already. I believe it's patented. Of course, you can also use for in the plant world, both not only detecting the gene expression level on tomato genome, at the same time, you can also look at the viruses infected by the in the tomato plants simultaneously. A couple of slides, 1 more minute, I'm winding up. We also got research solutions for COVID-19 more recently. We have free panel plus spike-ins. That's someone wants to talk. Okay. We have spike-ins, targeting 10 probes specific for COVID-19. We're giving this free for 48 assays. That when you buy any panels from NanoString, you can spike in. And so while looking at the virus, you can also look at the host genome and the impact in the immune response and other, if you're using cancer panel, the cancer genes expression level simultaneously. Probably Marshall will talk more about DSP solutions on COVID. These are the comparable panel for the free spike-ins that we're giving away. All of these panels can be purchased. And then when you purchase, we'll give you free 48 assay worth of spike-in COVID-10 probes for every single assay that you're running. Of course, a lot of people using these spike-ins together with our panel for therapeutic development and pathogenesis. Of course, a lot going on in the vaccine development and testing. There are a few publications within the last few weeks, high-end publication, NEJM, Cell, BMJ, fantastic papers. There are more in the pipeline. Watch out our website or talk to Bionivid or Ashish on that. With that, I'll wind up. But just to recap, the only technology, the qPCR has been around for close to 30 years now. The only technology right now does not use any RT-PCR and amplification and digital precision and highly multiplexed and simple and fast in detecting gene expression and other analytes. Thank you, and then happy to take any questions if you have.
Yeah, thank you so much, Jay, for such an informative presentation. And the technology looks really promising. We have few questions. I'll take it one by one.
Sure. Thanks, Rohit. Yeah, sure.
Yeah. So basically, this question is from Dr. Paritosh, wherein he asks, like, how sensitive, like, what is the sensitivity of the detection region of the probes?
The sensitivity region, you mean?
So, yeah, so
I think the question is
how sensitive these probes are.
Yeah, so we can detect all the way to 0.5 femtomolar. That's also our paucity control. If it's below, then we're not able to detect. So we can detect, if you have 100 copies, we can detect 2 copies of nucleic acid.
Okay. And
without
another
amplification.
Question?
But if you want, you can use our low input and amplify and count more if you want to. But without amplification, you can detect 2 copies out of 100 copies of nucleic acid in your sample.
Okay. Another question is from him only. He asks, how can we differentiate between homologs?
Yeah, so that's difficult because the probe will bind to both, right? In this case. Yeah, we need the way the probes are designed, it can, there's always variations, mutations and single nucleotide variations in the genome. So when we design probes, the probes, even with the SNVs, the probes still bind to the target. So we need enough variation for us to be able to design probe that differentiate 2 different transcripts. So in this case, our probe will bind to both and won't be able to differentiate unless there are ultra-specific probes that we can use to detect the variation. But it's complex and a little bit expensive than the usual way that we do. But there are ways we can detect even single nucleotide variation. But of course, it's in the normal nCounter standard assay that we don't do that. But that's the same technology that we use in our SNV assay. Even single nucleotide variation we can detect, but in general we cannot. The probes will bind to both homologs. So we need enough differences in sequence to be able to identify those targets. Okay. But then there are people doing isoform level. As long as you have an exon that is 100 mer different between the one exon, between one isoform versus the other isoform, then we can detect with the 2 different probes. We can specifically put one probe for one isoform and then totally different probe for other isoform. There are different kinds of things that people do. They want, in a single probe, they want every single isoform. And there are people asking a different isoform using to be detected using different probes. In some situation, in most cases, in a single probe, we can detect all isoform, but very rarely we will be able to put different probes for every single isoform. So, of course, it all depends on how different or how similar the sequences between the isoforms. And the same applies to homologous sequence as well.
So I just have question regarding the isoform. So
Yeah.
you would be able to detect the isoforms if you have a probe for it, but you wouldn't able to detect isoforms which are novel.
No, it's a good question. So if we don't know the target, we cannot.
Okay. Okay. Great. So another question is we have a lot of question on miRNA. So from the
Yeah.
Dr. Jyoti Singh, she
Yeah.
wants to ask how to capture the like does the probe work for small RNA like miRNA and piRNA?
Great questions. I didn't put this together, but I just want to quickly show. Hope I can quickly show microRNA. Just a second. It's
It was there. I think if you go up, it was there.
oh, it's there, is it? Yeah. It's easy with the cartoon, but
Yeah, slide number 8. You can. Yeah.
yes, it's not okay. Yeah, I think this should be fine. You're right.
Yeah.
Yeah. So it's a great question. Our probes are 50, 50, 100 mer, whereas microRNAs are super small, 18 to 22 nucleotide, right? So the question is, with 100 mer probe, how do you measure microRNA? Great question. I'm a big fan of nanostring microRNA product, the best in the world, this product. I'll show you. So we take every single microRNA. We only do for human, mouse, and rat. We take every single microRNA. And because the biggest problem with microRNA, for all the technologies available, they are super small, and putting a probe to target microRNA is difficult because it's small. The second problem, the difference between one microRNA versus the other microRNA in the same species or even any species, they're highly similar. Most of the time, it's just one nucleotide change. So designing a probe that specifically targets that microRNA is a massive challenge for all technologies. The third problem is microRNA is small, our probes is longer. How do we capture and then count? So what we did is we took every single one of these microRNA and attach a long microRNA tag sequence. This is unique and specific for each microRNA in the human. For a different microRNA, there will be a totally different unique microRNA synthetic oligo to be attached to this. We use enzyme ligase to attach this. So now this solves the three problems that I mentioned. Now the microRNA is longer. It's long enough that we can put capture probe and reporter probe and count them. That's problem number one. Problem number two, we know that between one microRNA versus other microRNA, they're highly similar. But by adding a long tag molecule, we ensure that each tag is completely different from one another. Previously, before adding a long tag, one microRNA versus other microRNA, just one nucleotide change. Now, after adding a long tag molecule, now the difference between one microRNA versus other microRNAs, it's massive. So now when you put the probe, it precisely specifically bind only to that microRNA because we are spanning a capture probe across this and then reporter probe across this. So what you're getting is a precise binding, highly specific binding between the nanostring probes against the specific microRNA. And then if you look at the microRNA, they're highly either low GC content or very high GC content. The melting temperature ranges very broad from 20 to 100. This is difficult for technologists to work with microRNA. So when we add the long tag molecule, we also control the GC content and the melting temperature. So by this way, the microRNA is long, and we ensure that each one is completely different from one another, so that our probes can not only bind, but specifically binds, and then basically count like RNA, count them using hybridize and count. No amplification in microRNA as well. Does that answer the question, Jyoti?
Yeah, I guess so. And there was another question from Dr. Shukla, but I think you have answered it in this slide, wherein he just asked that he used to add synthetic adapter to miRNA to detect 100 base pair in the technology. So I guess you have answered the question. How?
Really, that was the answer basically. That was not the question.
Okay.
Yeah. So Jay, can you just open the slide for PlexSet if you have, because there are questions of sample multiplexing as well, along with the target multiplexing. So just
I don't
one slide
have,
for
but basically you can combine 8 sample in a single run. You can so basically we have in the one cartridge you can run 12 samples. There are 12 lanes. But with PlexSet chemistry, what you can do, as long as your plex number maximum is 96, which means if you're detecting 96 genes per sample or below 96, what you can do instead of running 12 samples per cartridge, you can run 8 sample per lane, or in other words, 96 samples per cartridge. You can multiplex 8 sample in a single lane, like how you do with Illumina RNA-seq, for example. You can index them the same way. We use different barcode for same gene, 8 different barcodes for the same gene that goes into 8 different samples, and you can pull them. And exactly once you run the assay, we can deconvolute based on the barcode, and we know what gene. We also know which sample it came from. So basically, it saves cost and time, and you can run 96 sample per day. You can use this for COVID research. In 30 minutes hands-on with our COVID elements probe, you can do 96 sample at one go and get the result quickly. And we're talking about 10 probes, not just one probe like our qPCR people doing for COVID. Although we're not a diagnostic company, but if there is something, a research purpose, if people wanted to do, they can use PlexSet chemistry for COVID.
Okay. So another question again from Dr. Jyoti Singh, wherein she asked that how long it takes to make customized capture probes for novel mRNA or miRNA. So I guess you have answered the question.
Yeah. I think so we collect gene list, unless some people rarely send gene sequences because they want specifically the probe to be put on a specific site. And then within 24 hours, our bioinformatics designs the probes. Not the bioinformatically designs the probes, tells you where the capture probe and reporter probe comes into your sequence of interest or gene of interest. Are there any secondary structure? What's the GC content? What's the melting temperature? And then will the probe specifically bind or will to this site, or are they binding any off site? If so, what is the site and what percentage it will bind? Are there any cross reactivity? They'll list every single gene and the details of this. And then we let the customer to decide, whether you're okay with the design, or do you want to change the design, and do you want to remove some of those genes because there could be homologous sequence, they're binding somewhere else, or do you want to change the design to move around somewhere so that we can find the best spot specifically binds to that probe. Once the customer signs up, okay, I'm fine with the design, then we take 2 weeks to manufacture these probes. And I keep the 3rd week for shipping to India. So basically 3 weeks.
Okay. There is another question from Dr. Shukla. Can we detect, like, if we have a lower copy number as low as 3 or 2? And the technology detect that?
Not without amplification. That's where the low input kit we can use to amplify. And that's what you will be doing with any other technology available in the world. Like you're always amplifying and capturing those. We can do that with nanostring, but you will be doing similar to what you do with other technology. You're going to convert into cDNA. You're going to RT and enrich with the primer, and then capture with nanostring. Yes, you can do, but then you're going to use enzymes.
Okay. Another question from Dr. Paritosh. Wherein ask, what is the minimum number of RNA molecule can detect?
That's what I said, 0.5 femtomolar. So yeah, so input level. So you're talking about the lowest is, it's a tricky question. We recommend 50 nanogram with our Sprint system. But then the Nature paper I showed you is 25 nanogram on FFPE sample, detecting 355 genes, right? So I've seen people using 10 nanogram. The problem is you will capture high express gene with low input, but you lose the low express gene with low input. That's a trade-off. So what determines the ability to capture is the expression level of the gene, not really the technology. If you have high expressor within 10 nanogram total RNA, you should be able to pick. But if you have low expressor, you better to increase your input. So I would recommend don't go below 25. We can do single cell, but then we need to amplify. We can do with 10 nanogram. But then we need to amplify. So my recommendation is don't go below 25, because in a multiplex situation, you have set of low express gene, set of high express gene, and you want to capture both. So in that point, I would recommend minimum 25.
Okay. So there is a question from Dr. Sayak Ganguly, wherein he asked that my lab is working on environmental samples, microbial samples, basically. So
Yeah.
what is the efficacy of probes and assay for such
As many as bacteria as possible. In that case, we would have had like 50 probes targeting, few hundred bacteria in soil, for example. So the question is, you need to come up with the list of bacteria that you wanted to target. Then we can start designing probes. Unlike in sequencing world, you don't need to have any prior knowledge. You can just take the metagenome sample and run the sequence and find out what comes out, Versus here, we're going to ask you a question. Can you give us the list that you wanted to target? Do you have any idea what you want to expect in these samples? That's a problem. We need some input from you, versus you can do
So I guess we can also take as we do for metagenome, like just 16S RNA sequences from different bacteria, make a probe for it and identify that.
It's a great question. So this is, yeah. So when it comes to bacteria, absolutely, we can use 16X. There are a ton of 16X RNA, right? So you don't,
exactly.
This is the beauty. This is the beauty. For example, in human infectious sample, there is, 99% human host genome transcriptome. There is less than 1% bacterial, for example. Let's say if it's a bacterial infection, less than 1% bacterial transcriptome. Within that 1%, because there is ton of 16S RNA, it's more than enough to use nanostring probe without amplification to capture these bacteria at the same time as you capture the host genome. There are a lot of examples, papers, and this is a patented paper. What they did is a patient goes to hospital with septic shock, infectious, and he's going to die faster. He's in ICU. And the culture method takes days to find out what is the bacteria that he's infected with. It's basically E. coli in this case. Plus, you also need to find out any antibiotic resistance or not. Similar to what MedGenome came up with the technology. You can sequence and find out tuberculosis strain. At the same time, you can also find out antibiotic resistance, right? you can easily do with nanostring in a much cheaper, quicker, easier way. But I don't know why nobody's doing, but I wish I want to support someone doing this work. Basically, in this case, patient goes to hospital, they take the blood, and then deplete all the unwanted cell and lyse the cell and look at design. We already designed the probes targeting 16S RNA. And then we usually recommend overnight hybridization. In this case, they use 2 hours hybridization and then immediately run nanostring assay. Not only find the E. coli infection directly from the lysate, they didn't. You can also do this with extracting nucleic acid targeting 16S RNA. You don't need to extract. You can lyse with the QIAGEN RLT buffer and put the probes targeting 16S RNA from bacteria, and then find the presence. Plus, nowadays with the molecular signature, gene expression signature, you can find out antibiotic resistance. So which drug that patient is, resistant to, which ones are not, and then you can prescribe drug immediately. The patient goes to hospital within few hours. They find E. coli infection using nanostring plus antibiotic resistance.
Mm-hmm. Okay, great. So there is a question. Can we also detect fungal plant pathogen? So
Absolutely.
I guess yes, it's you have mentioned it earlier in your slides.
Yes, absolutely. It doesn't matter. Like I mentioned, as long as any genome in the world is sequenced.
Exactly. Yeah.
We do, yeah. In this case, they used bacteria, fungi, virus, and parasite, all 4.
Mm-hmm. Yeah. So again, a question on mRNA from Dr. Singh. So she is asking how it takes care of isomeric miRNAs.
Yeah, can you explain what's that?
Hello.
What's isom? Yeah, can you explain what's isomeric? I don't think I understand.
Dr. Jyot Singh, can you
unmute him?
Avinash?
Hello.
Yeah.
Hey,
Jay.
can you hear me?
Jay. Yeah, Jyoti is here. Yeah.
Yeah. So isomeric RNA, they differ only at the 5' end and for single nucleotide.
Okay. Yeah. So the MERBase 26 that has the 22 1800 microRNA, do they have isomeric? Is it part of
Yeah,
MERBase?
especially this new with the new RNA-seq technology, we generally get a lot of isomeric RNA. And since there is a change in the 5' end, so that the targets are different. So they target different genes.
Oh yeah. So I get it. So that's why the same microRNA with the _5 or 3, right? Is that why when the naming microRNA, because they're 5' or 3'? Is that right? Is that how it works?
5' because so if they differ in the 5' end, definitely they will go and target some.
Oh yeah.
different-
So yes, the answer is yes.
Okay.
So
and
mostly.
Yeah, I guess Jyoti here, like, as long as you have the sequence, you can design a probe, whether it is different from at 5' or 3', we can sequence it.
So
I
no,
guess
my
so.
only concern is the ligation steps. So as we know that ligation steps is very ambiguous because when we do even for 5' or 3' race, so again the use of ligases are mostly
Biased.
ambiguous.
Okay.
Yeah. So
Yeah.
how
You are
Yeah,
talking about the biasness to in the ligation step.
Ligases have and then since because the extra step we do for small RNA, as I understand, is that you do a ligation of all the small RNA. So first is the enrichment of the small RNA, and then you add the your extra synthetic small RNA sequence with all the microRNA, and then you will make a probe based on that additional sequence, right?
which all fits into one temperature. So we have 5 different, each goes into 1 minute and PCR machine. ligates different microRNA at different temperature. But after 5 minutes, all microRNA would have been ligated to the tag. Yeah, you're right. So exactly same protocol, but just the 5 different temperature to making sure every one of those microRNA are ligated.
And one more query. So, since you mentioned that your microRNA probes are only restricted to human Drosophila and
Mouse.
Mouse.
Human mouse, right? For now,
So
yes.
Can you also customize it for other insects? Like I work on silk moth, so can you customize your?
No, I get this question on a daily basis for plants as well, because microRNAs are everywhere. The problem is, as you notice, there is microRNA, and then there is a tag molecule, and then there is capture probe, reporter probe, and each species, when we use for human, it's repeatedly ordered the same, whether it's custom or panel, the same design is being applied, used. We cannot stock for everyone, order oligos, design, and test every single microRNA separately. And then we pull them and do QC as the whole code set together and QC before shipping. And doing this for each custom project, it's a lot of work unless the study is very big. Doing it for small scale projects, a lot of R&D work and QC work. And then the inventory is not useful to anyone, because the species that you're working, the microRNA that you're interested in, not so useful to anyone else probably. It's a very unique project. So we decided to work with the most common genomes that people often buy versus plants or other species.
Okay, great. So there's another question from Dr. Sachin Kumar, wherein he asks, is it possible to detect expression level of multi-copy genes located on different chromosomes in a genome?
Which means the same gene sitting in different side of a chromosome.
Yes.
So the probe doesn't care where they're sitting. As long as they're complementary, they will go and bind, and you get counts. Yes, the answer is yes.
Okay.
This is the exact problem. This also poses cross hybridization. When you when you design a probe that is also complemented to somewhere else, the genes on the others somewhere else in the genome, that's a problem, right? So it's an advantage when you want to do this kind of project, versus it's a disadvantage when you want the probe to be specific.
Yeah, so actually, Jay, most of the places people look for like chromosome level, different gene copy numbers at different locations. So instead of like, if you design a probe, it will go and hybridize to the RNAs from all the copies. Is there a way to segregate based on the chromosomal copies?
No,
it's like different paralogs things.
Yeah.
So yeah, the same with RNA what we have. Like,
so
in RNA, it becomes difficult to do the copy number.
So the gene sequence is precisely same, but they are located in different regions. Then it's difficult.
Yeah.
Unless there are unique regions within the gene that are differ between one another, then we can precisely put the probe where they differ. But otherwise, you don't know where the count's coming from, whether the chromosome 1 of that gene or chromosome 5 of the same gene. You won't be able to tell.
Yeah, mostly what I see, if this reason, like gene, is coming from different chromosome, there has to be some difference in the sequence. Like the simple, like what I'm doing right now, the project for the cotton. Love, you already did almost 192 sample for the customer. So that plant has A and D different genome basically, and they were not sure whether this gene is coming from A or D genome. Okay. The good part was there was a difference of 3 to 4 base pair with each gene sequence. So we were able to design the probe, and we were able to count that molecule.
Yeah. Any other questions? So if not, I think we should let Marshall.
Yeah. Okay. So I guess that is it. I hope I didn't miss out any of them. So thank you so much, Jay, for answering, taking up all the question and answering beautifully. Thank you.
Yeah, thank you. Thanks so much.
All right, can everyone hear me and see the screen?
Yes.
Yeah, we can see.
Okay, all right, great. All right, thanks, Jay. Hello, everyone. And just briefly about myself, my name is Marshall Fettrell. I'm the technical sales specialist for the Asia Pacific region with Nanostring. I've just recently joined the company about 2.5 months ago. But I look after our Genomics digital spatial profiling instrument. So I'll walk you through a presentation of the technology, give you a couple examples from the literature, and then field any questions that you might have. So as Jay has mentioned, he was talking about bulk genomics, and obviously we know the importance of that, and Jay provided many examples of that on NGS and also on our nCounter system. But we know from 2006 on that there's been a transition from bulk genomics to single cell genomics and then over to spatial biology, which I'll be talking about mostly today. So with bulk genomics, obviously we can answer important questions, but the difficulty when you homogenize a tissue or a sample is that it's difficult to map that genetic signature back to what cell that it actually came from. And so with the advent of RNA-seq, which is a tool that Jay had mentioned, and obviously 10x has done a good job with that, now we have this multi-omics approach to genomics where we can actually identify what cells are expressing what, and we're able to dissociate tissues and identify important cellular subsets that we weren't able to do previously using bulk genomics. But of course, when you do move into single cell biology, you're missing an important spatial context for how those cells actually align within a tissue, how they speak to one another, and how they're interacting. And we know, particularly, in the cancer, for instance, that the tumor microenvironment, that those cells together, that they're not all passengers within that environment, that they all speak to one another, and that shapes disease progression. And we'll be able to understand that. So last year we launched this Genomics digital spatial platform in April of last year. So of course, up until recently, there was a paradigm and a trade-off between looking at spatial versus high-plex experiments. So on the left, sorry about that. So on one side, you have high-plex expression profiling tools. So that's your NGS, the single cell, as we've mentioned, and flow cytometry. And on the other side, you have imaging. And up until recently, there was no really option to integrate the two. So at Nanostring, we thought we could combine and leverage our existing expertise in bulk digital molecular quantification using the nCounter system, and then combine that with imaging so that we could look at both RNA and protein on a singular platform. Of course, the challenge with heterogeneous tissues is the biggest portion of it is often not relevant to the disease being studied, and it will simply dilute the signal that might reveal difference in expression levels, for example, between two patient groups. So in this example, if you assess these two groups using bulk methodologies, using NGS, for example, you homogenize the tissue, and the genetic signatures will be similar as you see here on the left. So if we focus the analysis of a smaller piece of tissue and to a piece of tissue that you find to be biologically relevant, we'll be able to determine differences in genetic signatures and differential expression across those two tissue segments. So as an example of this is a study that Nanostring that we did in collaboration with Merck, where they were looking at the effects of an anti-PD-1 blockade in a patient cohort. So they took FFPE tissue samples collected from these patients, they ran them on the nCounter platform, and what they were trying to do is identify a genetic signature that could predict response to an anti-PD-1 blockade. So if we look at the graph here down on the bottom, on the x-axis, we have something called a GEP score, which is what we call at Nanostring called the tumor inflammation score. And on the right, on the y-axis, we have progression-free survival. And this GEP score, or this TIS score, is an 18 gene signature, which was identified on the nCounter platform, and that showed to have correlation with response to anti-PD-1 blockade. So on the left hand of this graph, they found that in non-inflamed tumors, and those patients that had non-inflamed tumors, that they rarely responded. And on the right side, almost all the responses came from these hot tumors. But within this type of bulk analysis, we couldn't differentiate between those that responded, and on the bottom here, in the right, there were certain patients that didn't respond, and we were unable to resolve that. So that we know, if we look at a tissue section of tumors, we know that some biology is simply spatial, and it cannot be resolved with that bulk analysis, as I mentioned. So here you have 2 tissue samples that have been analyzed by gene expression, and they calculated the TIS score that I mentioned, which is the tumor inflammation score, which is high in both cases. But when you look at the left piece of tissue, you can see that the green, here's a pancytokeratid stained tumor area, is devoid of immune cells that have been stained in pink and blue. Therefore, this is a hot tumor, but it's an excluded tumor because we don't have any immune cell infiltrates. So in contrast, in the right tissue sample, you can see many immune cells directly within the green tumor area, making this a highly infiltrated tumor. And so you can imagine that these 2 patients would require a totally different treatment modality. So in the case on the left, the immune cells would have to be drawn to the tumor area, while in the case on the right, the immune cells are already at the site of action, but they might require an immune checkpoint blockade to fight off that particular tumor. So in terms of spatial context, what we need is a platform, and what we designed is a platform that could identify a lot of different metrics that are very important for researchers and for translational research as well. So we needed something to detect all targets with a degree of high sensitivity with unlimited target detection. As Jay's already gone over our nCounter detection chemistry, we utilize that on the DSP platform in addition to our new NGS rollout. You want to be able to assay any analyte. So we have validated content to detect both protein and RNA content. That's both on an nCounter readout and also for NGS. And you'll also want to be able to explore any region of interest. So as I mentioned in that example before, you want to be able to look at a tissue section, find the biology. It's important to you, and be able to profile that accordingly. You also want to be able to access any sample. So you don't want to be limited to just FFPE or fresh frozen tissues. You want to be able to do both. On the DSP platform, we have a tremendous amount of flexibility for what you can use in your samples. So FFPE, fresh frozen tissues from mouse, from human. We can also do tissue microarrays, punch core biopsies. Any of those types of samples can be used on the platform. And then finally, you want to be able to trust all of your data. So we have a whole list of publications. We have optimized validated content for our antibodies. So all of you know that can take time, weeks to months, to validate a high degree of multiplexing antibodies on a particular section. And then you want an automated solution as well. So how does the technology work on the DSP instruments? Jay has already mentioned about the capture technology that we run on the nCounter. But for the DSP, we have 2 offerings. So you have protein and or RNA. So for protein, we have validated monoclonal antibodies. They're covalently linked to this DNA oligo that Jay has mentioned, and it's attached via UV photocleavable linker. On the RNA side of things, we also have an ISH probe that sits along your target RNA, and that's attached to a DNA indexing oligo as well via this UV photocleavable linker. And so within this assay as well, what we have spiked in is what we call morphology reagents as well. So you can stain your tissue section with up to 4 targets. So 3 of them are just fluorescently labeled antibodies, and the 3rd for a DNA stain. And if you look over here on the right side panel, we put the slide into the Genomics instrument. It takes an image, and this is in 4 channels. So we have FITC, Sci5, Sci3, and Texas Red. And then it puts that image, and then we visualize that image, and then we use those morphology markers to define regions of interest that we're looking at. So within those regions of interest, we're able to profile any analyte that we're looking at, and then we get a particular readout. So here we have a heat map. So once we start to zoom in on a region of interest that we want to profile, so here in the middle panel, you can see that you have a labeled FFPE slide with our probes. These are DNA oligos with our UV photocleavable linker. We shine a UV light on that tissue section. And what it does is it releases those barcodes into a buffer system. And then the instrument is able to sip that buffer and those barcodes up, and then it deposits it into a 96 microteter plate, 96 well microteter plate. And as I said, we have a high degree of multiplexing capability with this, so we can profile up to 96 proteins on a particular slide with the nCounter. We can also profile up to 84 RNA targets using the nCounter. And with our RNA NGS readout, we can multiplex up to over 1,800 genes with and our whole transcriptome offering, which is coming out later in the year, up to 18,000 genes. So as I was just mentioning there, here's the Genomics instrument here. So with your samples using either, depending on what content you're looking at, either protein or RNA, that can be read out on the nCounter platform, which Jay has covered, and or on a next generation sequencing instrument. So if you have any of these instruments in your core facility or service facility, you can run any of those NGS readout. So here's just an overview of a little bit about what the workflow is and what it looks like. So in terms of imaging reagents, as I mentioned before, we use these as morphology markers to help guide our region of interest selection. So you can use any antibody. These aren't proprietary to NanoString. If you have a working antibody that's tagged to any fluorophore that works in the channels that I mentioned, you can use those as markers to identify your regions of interest. We also have a collaboration with ACD Bio, and you can incorporate RNA scope probes into our NGS readout assays. So if you have a particular antibody that's not performing well, or if you have a particular cell or transcript of interest, you can identify those cells using RNA scope probe, and then we can use those to identify regions of interest. And again, you have your profiling reagents, either your RNA or protein content. You bathe your tissue slide on that overnight, and it's stained and ready to be run on the DSP instrument. So how it works, as I mentioned, so you'll stain your slides, either protein or RNA, and then we use those immunofluorescent tags to look over the tissue section and identify a region of interest that you're interested in on the tissue section. You can do up to 96 on one particular slide in an experiment. Once you define those regions of interest, again, you shine that UV light, and then those barcodes are sipped up into the instrument and deposited into a microteeter plate. And then you'll go through and run a counting sequence, either on your nCounter or on your NGS filing. So how does that work? So the DSP has these adaptive optics, which is called a digital micromirror chip. So these are 2 million chips that are on the bottom of the sequence, and we're able to configure these mirrors in a way that they can actually trace very complex and unique biology. So if we look at this example here in the middle, this is a tumor section right here from a TMA. In yellow here, this is a pancytokeratin stain of your tumor cell, and the sort of purple magenta color is a DNA stain. And what we can do within this, using this sort of segmentation or masking strategy, is that we can identify all of those cells that are associated with the tumor using this tumor mask. And then we shine the UV light on this particular area, and then we'll profile all of those analytes that are interested in that particular compartment. And if we flip that mask over here, you can sample everything out of the tissue microenvironment. So further to that, we have a tremendous amount of flexibility in terms of how we can profile these regions of interest. So when you're defining a region of interest, as I mentioned before, we can use these different geometric shapes. So either a circle, a square, or you can use polygons. And then within these geometric shapes, you can do other forms of profiling. So you can do a segmentation profile. So this relates to the example that I gave before, where you're able to segment out a tumor in the tumor microenvironment. We can do cell type specific profiling, where if you have morphology markers that identify specific cell types that you're interested in, we can put a mask over those cell types and then profile from them accordingly. We can also do contouring strategies as well. Within that region of interest, for example, here we have a tumor, tumor's on the inside, and here's a feature of interest. You can look at the invasive margin along the tumor, or along an astrocyte, or whatever type of biology that you're interested in. Or if you're looking for a complete unbiased sort of discovery kind of project, you can use a gridded approach and sample across the entire section. So here's just a case study, an example of this. This is from a recent study from David Rim out of Yale University. This is the third publication that he's done on the Genomics instrument. He was one of our beta users. So in this particular paper, this was published in the Journal of Clinical Cancer Research. And so just some background on this particular study. So the majority of patients with advanced non-small cell lung cancer, they do not respond to PD-1 axis blockade, particularly as a monotherapy. And since PD-1 checkpoint blockade is a standard of care for advanced stage disease, they were looking for more indicative biomarkers of discovery. And then further to this, they're looking the need for identifying more predictive biomarkers, because what they've found is that, and studies have shown this, there's poor intra-observer reproducibility of PD-1 scoring using IHC alone. So their experimental design was the following. They used biopsies from 53 advanced non-small cell lung cancer patients, and they were taken prior to treatment with a single PD-1 blockade. These were done in TMA samples. And then what they looked at was a large panel of 44 antibodies used to screen each of these for potential biomarkers that might predict susceptibility to the anti-PD-1 blockade. But not, so, and then in terms of their profiling strategy, what they looked at is measuring these 44 proteins in 4 different tissue compartments. So they looked at the tumor, macrophage, total immune, and non-immune stroma. And then the expression of all these markers was assessed. Okay, so on the left here, this is what we have. This is one of our, the initial fluorescent image that was taken of the tumor cell. You have pan-cytokeratin in green that identifies your tumor cells, CD45 that identifies your immune cell populations, and then they put on a CD68 to identify macrophages. On the right here, you see this, excuse me. On the right, you see a segmentation profile that they put onto this particular segment. So for the tumor cells, were identified by that pan-CK. You have in yellow CD45 compartment for your immune cells, CD68 in blue, and then they also used, they looked at a non-immune cell stroma compartment, which is this purple segment that sort of bifurcates the sample. So the primary finding that they identified were 2 biomarkers that corresponded significantly with all clinical markers, including both progression-free survival and overall survival. And so that was CD56 and CD4. And they found that these could potentially be used as biomarkers. So what I've not shown here on the slide is the high levels of VISTA and CD127 proteins in the tumor compartment were associated, excuse me. Sorry, so VISTA and CD127 proteins in the tumor compartment were associated with immunotherapy resistance. And this, that was for both, was an indicator of shorter progression-free survival. So in essence, that served, those 2 markers served as a negative marker for successful therapy. So in this instance, both CD that's extremely important in this study. So now I'm just going to switch gears quickly and just show you what we have on offer and a few more studies as well. The DSP instrument, we have over 140 targets for cancer and immuno-oncology. How the panels are selected is you choose a 20-plex, well, you have to choose a human protein or a protein core. It's 20-plex and it has the housekeeping genes spiked in. And then you can choose up to 6 10-plex modules as well. Here's the content here. So you have immuno-oncology drug panel targets, immune activation, pan-tumor models, some signaling modules, cell death modules as well. And then we have a space for customization as well. So if you are interested in developing custom markers, we can do that for you. And we also have a partnership with Abcam where we can do that. So again, this is up to 96 plex. For neuroscience, we have over 75 targets. And we also have content for immuno-oncology for mice, and in neuroscience for mice that'll be released later in the year. We also have plans to do an NGS readout for protein later in the year. On the RNA side, for readout on the nCounter, we have what's called the immune pathways core, and that's up to 84 genes. That covers a global immune response, and that response is important for the tumor microenvironment in the tumor. and it also encompasses the tumor inflammation signature and other signatures that we found useful in our nCounter panels. So this is the latest offering that we're going to have released at the end of July. This is our Cancer Transcriptome Atlas. So this covers over 1,800 genes across 55 pathways, and it relates to tumor biology, immune responses to the microenvironment, and can identify low expressors as well. I'll talk to you about the chemistry and how we're able to do so. This is all built off the back of our successful nCounter panels as well. So this is done in collaboration with researchers, and it's shown over the literature, as Jay has mentioned earlier. You have the ability to customize probes with this offering as well, so you can spike in up to 30 genes. The sequencing requirements for this is 30 to 50 million reads per sample, or that's per slide. That can also vary because we sequence according to area, so there is some variation there, but we can talk about that offline if people are interested. So in terms of the chemistry, we have an ISH probe, and we do all of our targeting in situ. So here you have your RNA target that's attached on the slide within the tissue. We have our RNA probe. We have 5 probes that actually sit along each of the mRNA transcripts. And so when we're running through our QC metrics and running these on the NGS platform, we're identifying 5 probes across the back of that mRNA transcript. So we have a tremendous amount of sensitivity. So attached to our ISH probe is a photocleavable linker, as we've mentioned before, and this is what the total library, or the total probe, looks like. So we have adapter sequences for your Illumina sequencing indices. So we have indices for the area of interest, or the region of interest that actual transcript was taken from, and then it will also add your P5 and P7 adapters, so you can attach those to a flow cell. We have a UMI sequence of 14 nucleotides. It's done to account for any PCR amplification. We have a single PCR amplification step in our library prep. And of course, you have a unique probe ID that can be read in your NGS readout. So this is an example of some of the analysis modalities that we're looking at. So on the left, we have pathway analysis. This is something called a joy plot, where you can actually look at the region of interest, and you can look at genes expressed around it. We have something called a reactome profile, which is down on the bottom left here. Actually, maybe that might help. There we go, reactome. So you can look at signal transduction pathways. You can look at other different pathways. This is all interactive, and you can highlight these different spindles, and they'll map back to that tissue sample. On the right, for biomarker discovery, we can do contouring segments across the invasive margin of a tumor. I'll talk about an example about this in the next slide. Within these compartmentalizations, you can look at volcano plots. So on the right, you might have your the tumor microenvironment, the tumor itself, and you can actually visualize expression profiles within those two compartments using these volcano plots. And we also have a quantitative single cell deconvolution method that we're developing, so that you can actually make use of your RNA-seq data and integrate that into what you're seeing on the DSP. And I have an example of that later as well. So here's an example of our Cancer Transcriptome Atlas that was run on a colorectal cancer sample. On the left here, you can see a tissue section of that colorectal cancer sample. In green here is a pan-cytokeratin stain for the tumor itself, CD45 for our immune cells, also CD3 here in blue. We used a contouring strategy along this tumor, so we can identify different features of interest along these segments of the tumor. So on the right here, in terms of interferon gamma signaling, you can see that within the tumor, that there's higher expression of interferon gamma as you move farther away. That signal lessens. For chemokine binding, you'll find the opposite. There's not much expression of chemokines, obviously within the tumor, but on the tissue, tumor microenvironment outside. And then for MHC class 1 and 2 antigen presentation, you can see that there's a high expression of those markers within the tissue and along the invasive margin as well. Coming out, available through our Technology Access Program, and in Q1 of 2021, we have a whole transcriptome atlas offering. So this is a comprehensive content of over 18,000 genes, and this is a highly curated content. So when you're sequencing and using a poly A tail grab, which we don't do, you're sort of wasting your sequencing dollars because you tend to capture the high abundant expressors. So we did everyone a favor and we removed the top 10 expressors out of our profiling. So most of those are beta-actin, microtubules, et cetera. And we also took out mitochondrial genes as well. So you have the ability to spike those back in to this assay if you want to, but typically most of your sequencing dollars are thrown away with that. So we've been able to help you out there. You can spike in genes to this, as I mentioned, and the sequencing depth for this particular assay is 100 million reads per sample. It's a guideline. So here's an example of the whole transcriptome and what this can look like. So again, I showed you this analysis sort of too earlier, this joy plot. So within this joy plot, you have your regions of interest that are within here. This is a fibroblast section. So each of these small spikes here that you can see in different colors, so you have purple and blue, each of the spikes represents a particular gene. And you can see these segments that sit around the circular section. And these correspond to different pathways. So now you have this sort of visualization tool where you can identify what particular pathways are being activated in that particular region of interest. And this is all completely interactive, so you can highlight a gene, it will map over to a heat map, and you can look at how these particular signatures look across the tissue and whether or not you have differential segmentation from different markers. So this is an interesting example. So this is from Alex Swarbrick's lab at the Garvan Institute in Australia. And what Alex is doing is he's developing a breast cancer cell atlas. And what he's done is a lot of interesting work using single cell technologies. And Alex is trying to profile what he calls the tumor ecosystem. So that's a conglomeration of neoplastic cells, of immune cells, of epithelial cells, perivascular cells that comprise the tissue. And he's been able to dissociate those using single cell technology to identify these particular cellular subsets. So in this experiment, he used our whole transcriptome atlas, and he was trying to map these signatures back to what we were seeing on the DSP. So on the DSP instrument, he used three different morphology markers to drive his segmentation profiling strategy, pan-CK for the tumor, and then he used a CD3 positive and CD3 negative compartment profiling. So if we look at on the right here, you can see on the top, these are all of the signatures and gene signatures that were coming out on the whole transcriptome atlas. He was getting anywhere from 7,500 to 9,000 genes, 8,000 genes per region of interest. So a tremendous amount of throughput that he's finding through there. On this axis right here on the y-axis, x-axis, the red here is a CD3 positive compartment, the CD3 negative compartment here in the green, and this is the tumor compartment here in the blue. And what he did is he mapped back his single cell RNA-seq data here on the bottom, and then map those signatures back to what he was seeing on the DSP instrument. So in the cancer section here, you see a lot of cancer epithelial cells, which makes sense, some T cells and B cells. And then what he found is a high concordance of those single cell genetic signatures here underneath that map that back. So this is just a useful way that people are actually seeing, mapping back that single cell RNA-seq data into space. So for this NGS readout modalities that we have on the DSP instrument, those can be run on any of these Illumina platforms that you may have. There's not the HiSeq is not in here, but it can be run on a HiSeq instrument. So if you have any of these in a core or service provision, you can use your cancer transcriptome atlas, the whole transcriptome, and as I mentioned, protein will be run on an NGS readout on protein will be coming here and fit on any of these instruments. So as Jay alluded to, we have content for COVID-19. Obviously, the pandemic is affecting all of us. So the current research paradigm for COVID-19 is falling into these 4 bins. So we have a molecular response, cellular response, tissue damage, and then individual genetic susceptibility to the disease. And we're trying to address all of those on the DSP. And this is coming from demands from our customer base. So for COVID-19, it's the offering that we have is in 2 tracks. On the protein side, we have a series of fluorescent antibodies that are available from Abcam that identify the virus and also important nasal and lung epithelial markers. And what we have is a custom targeted panel that's being released by Abcam. It should be out in the next couple of weeks, actually, which you can pair with our human immuno-oncology profiling panel. So you select your core module, you can select up to 6 10-plex modules, and then add this custom targeted panel for COVID-19 in there. On the RNA side, we have a partnership with RNAscope, as I mentioned earlier. So RNAscope has a series of probes that are available for the virus itself, for the ACE2 receptor, for the TMPRSS protein, and for important cytokines in the host immune response to the virus, and obviously for the cytokine storm. The offering, excuse me, this is not behaving. Yep. So the offering that we have for this right now is called the COVID-19 Immune Response Atlas. So this is built on the back of the Cancer Transcriptome Atlas I mentioned. So there's close to 1,900 genes that are available. This covers all the immune response plus targets for the virus itself and critical host genes important for fighting the infection. And the plan for this, it's not going to be commercially launched, but it's treated more as a customized type of offering. And it will be available through our Technology Access Program up until the end of the year when we release our whole transcriptome atlas. And once the whole transcriptome atlas is released, if you are doing COVID-19 and using this particular product, we can spike those genes back in. And so here's just a more detailed outline of the targets that are available for the COVID-19 product. So those specific for COVID-19, for COVID-19 receptors, alveolar markers, lung biology markers, viral response, and other important genes associated with interferon gamma signaling and myeloid activity as well. Again, you'll pair those with RNAscope probes. As I mentioned previously, all of these are available through our Technology Access Program. So this is run in our R&D lab or through our TAP lab in Seattle. So you'll send us an FFPE sample, you select the particular content that you're interested in. This is a highly collaborative process, so you'll work with myself and our team in Seattle to select your morphology stains and profile the areas of interest that you're looking at, and we send a report to you. And this is used as a proof of principle concept. On the data analysis side, we have a very integrative software suite of tools. So for the Genomics Control Center, this is where you select your regions of interest across your sample. You can prep either your NGS or nCounter library. That's brought back in and converted into count files if necessary, and then you can view this on our data analysis suite. And our software is actually pretty exciting. So on the left here, this is just a visualization of our analysis suite, so you can actually look at the tissue itself, and then you can use all those different visualization tools that I mentioned earlier. So volcano plots, you can look at box and whisker plots, etc., heat maps, and you can highlight these different areas and it will show you exactly where that is on the tissue. And you can also analyze multiple tissues at once, so it's pretty slick. So in terms of how it's implemented, we have a cloud architecture, which allows many users to interact with the system at once. So while only one user can actively control the instrument at a time, many users can be aligning images and selecting regions of interest to profile or analyzing their data simultaneously. All right, in terms of publications, we have quite a few. They're coming out rapidly. We have 19 peer-reviewed publications to date, including cover publications for clinical cancer research. We also recently had back-to-back publications in Nature in January of this year. All this information is available through our website, or you can reach out to myself or Ashish, and he can send this to you. So just to wrap it up, you can multiplex, you can do many analytes on one tissue slice in a single pass, and then you can analyze multiple analytes, both protein and RNA, and you can do this quantitatively with high resolution, and it's not destructive to your tissue sample. And then in terms of translation into the clinic, you can do up to 12 sections per day, so it has that sort of clinical variability. All right, so with that, I will answer any questions that anybody may have.
Yeah, thank you, Marshall. There was a question, I think, which has been taken up by Jay and Ashish. So I have a certain query. Uh, Basically, you have mentioned about immunofluorescence tag. So, are you
Yes.
preparing these fluorescent tags for each cell type?
Yeah. So as I mentioned, so the morphology markers that we have some like pre-canned content, but it's for essentially for solid and solid tumors and melanoma. So those are typically like CD45, pan-CK, SM100B, etc. But you're not limited to those. So you can choose any antibody that you want, as long as it works. It's conjugated to an immunofluorescence tag. You can use that to guide your assay. So we've done some projects on different tissues through our TAP program, and we have some of that available and stuff that we can share with people as well.
And how sensitive are these tags? Because a lot of times we face this issue, like while doing wet lab also, fluorescent tags, you get a lot of noise. So how sensitive are these tags?
Okay, so that's more on the detection level, you mean? Yep.
Yep.
So it's actually, so our detection chemistry is not related to fluorescence. So if you remember, we're actually capturing the barcode itself. So the limit of detection on the protein side, it's not actually limited by our chemistry. It's actually limited by the number of antibodies that you can put onto a slide. So if you can imagine, at some stage, you're going to get some sort of cross reactivity or saturation just with, with too many antibodies being placed onto the slide. In terms of NGS, really the sky's the limit. So we don't really have any kind of limit to how high we can plex.
Yeah, but binding to antibodies also, when antibodies also have binding, you get a lot of noise, right? Antibodies are not very, very specific. So how you deal with that?
Well, yeah, that's true. So yeah, at the moment, I mean, we're limited to 96 plex.
Okay.
At the moment, and that's where, so we have, if you're looking for validation, so we validated all of this in tissues. So we've cross-validated. We actually just had a recent paper published in Nature about this, like validating our protein and NGS content together. So we validated all of that versus bulk methods and other IHC methods, and found high concordance with plexing up to that level.
Okay. And I was just wondering, is this technology sensitive enough to detect the methylation pattern?
No, I don't believe so. Jay, can you comment on that? I'm not actually sure. I haven't heard anything about
No,
it.
I think we can't do with that.
Yeah, we can. If it's not bisulfite conversion, we can. But if it's bisulfite conversion, we cannot.
So without bisulfite conversion, you would able to detect, is it?
We can,
because I guess there is some.
We can pull down, like we can do chipsticks and things like that, but
not real time detection, because okay, fine. And I was wondering this about like circulatory cell-free tumor DNA. So how sensitive this technology is to detect?
Yeah, you probably have to amplify, because there are not enough RNA molecule to do without amplification. So at that point, you probably need preamp kits. We can do low input using nanostring. So you need to amplify. Otherwise, it's too few in some situations.
Exactly.
To detect.
You need low input. I mean, see, these tumors cell-free DNA will be present in very low quantity in the cells, right? So
yeah,
if you're taking a very low input, are you not missing on that?
No, what I meant very low input is the amount of the circulating RNAs in the circulation. It's very few. In that case, to detect, you need to amplify, increase the counts, enrich them basically, and before you can hybridize with probes.
But most of the cases in circulatory tumor RNA, people look for the mutation information, not for
Yeah.
maybe the expression values. So definitely that's not a domain which where we work.
Mm-hmm. Okay.
Yeah, a lot of people do digital PCR, droplet digitals with the.
So single cell people are working, like single cell RNA
Yeah.
isolation. People are working with the circulatory cell-free RNA.
That's
Okay.
right. Yeah.
Okay, fine. So I think that is it. No more questions. Thank you,
Yeah,
Marshall.
I think one question, Roli, I think one question I would like to put it to Dr. Vishal has asked for this nematode one. Basically, I want to get it answered by Marshall as well.
Sure.
As per my information, I think we have not come across any example as of now. The technology is also new. But what to say, Marshall? Can we, like, if somebody is working on nematodes like C. elegans or something where this worms is there and it's very, very transparent, so whether we can utilize this DSP technology for that kind of work?
Yeah, so I mean, at the moment, we've primarily been focused on human and mouse examples for particularly for the nCounter readout. But I think, as we sort of transition over to a more discovery market with the whole transcriptome, that might be something that we'd be able to look at. But at the moment, there's no examples of that using this particular technology yet. But I'm excited to see, we could, potentially, I think that could be on the horizon. But
yeah, section, I think making a section is not a problem, but I think if you have some visualization markers, because first we have to see what are the visualization marker, or because I don't see any RNA scope probe will be there for nematodes. So definitely we have to look for any protein marker only, antibody based marker. So Dr. Vishal, if you have some like marker which you identify that these can give you the cell surfaces, basically that this kind of cell types is there. But definitely if you make a slide and you have those markers and it could be tagged with our synthetic oligos, definitely we can work for nematodes as well.
Yeah.
But as of now, nobody has done it. Yeah. I got a reply. Thanks.
Okay. Thank you, Marshall.
Thank you very much, everyone. Thanks for sticking around.
Yeah, sure.
Yeah.
Hello. Hello.
Hello, we can hear you.
Yeah. Like, so on behalf of Bionivid
Hello,
Technology.
yes, Rohit.
Yeah, Rohit sir. Hello.
Yeah, hello. Yeah, tell me.
Yeah, I think Mr. Rohit was about to speak in between.
No problem. I think thank you so much,
Okay.
Bionivid and all the customers who have participated in this webinar. We want to thank them. That's it. Yeah.
Yeah. Okay. Thank you so much.
And for any query, they can reach out to you and or us anywhere.
Sure.
Yes, so thanks to the Leverage team and all the speakers and the participants. And we will definitely take it forward. And we will get to you if more queries that we get from scientists and all, and we will get back to you for more clarification. And like we had around 150 plus registration, I think many people could not join. So we will share the recording and get more feedback. And definitely we look forward to take this association further. Thanks again to the whole team.
Sure. Thanks, Rohit. Thanks, Rohit and team. Yeah.
Thank you.
Thanks, everyone.
Thank you.
Thank you, everyone.
Roli, Avinash,
Thank you, everyone.
for coordinating it.
Have a good day.
Thank you.
Bye, guys.